Imagine for a moment holding the instruction manual for human life, a complete blueprint for, well, everything, from the color of your eyes to maybe your predisposition for certain diseases. It sounds like something straight out of a science fiction novel, right? But what if we told you that scientists have already done just that, meticulously mapping the incredibly complex genetic code that makes us human? Today, we're embarking on a really deep dive into one of the most ambitious scientific endeavors in history, the Human Genome Project. We'll unpack its audacious goals, the incredible technological leaps that made it possible, and, the profound, sometimes unsettling questions it continues to raise. Questions about our identity, our health, and the very future of humanity. We've pulled together a stack of sources, scientific papers, historical accounts, all ready for us to distill. It's kind of a shortcut for you to really get a handle on this monumental achievement and its ongoing impact. As we journey through the project's origins, its breakthroughs, and its lingering ethical dilemmas, we invite you to consider, what does this intimate knowledge of our genetic code truly mean for us? Both as individuals and as a global society, what responsibilities does it place upon us?
That's really the core question, isn't it, that anchors this whole conversation. The Human Genome Project, it isn't just a scientific accomplishment. It's arguably one of the defining turning points in how humanity understands itself and its place in the biological world. It fundamentally altered our approach to biology and medicine. It really set the stage for an entirely new era of discovery, one that continues to unfold right before our eyes.
I couldn't agree more. The sheer scale of it and the vision, it's just astounding. So let's start right at the beginning. This wasn't some isolated lab experiment, right, done by just a handful of researchers. This was, and still is, hailed as the world's largest collaborative biological project. So what exactly was the Human Genome Project? How did something so ambitious even begin to take shape?
Well, the Human Genome Project, or HGP as it's widely known, it formally launched back in 1990. It was planned as a 13-year international scientific research effort. Its primary objective was, well, remarkably ambitious, almost audacious for the time. The goal was to determine the complete sequence of the estimated 3 billion chemical base pairs that make up human DNA. And alongside that, a parallel goal was to identify and map all of the genes within that sequence. At the time, they thought it was roughly 30,000 genes. And to give you a sense of the global scope, this wasn't just a U.S. thing. It involved scientists from 20 universities and research centers spread across 6 different countries. We're talking the U.S., the U.K., Japan, France, Germany, and China, all working together.
Wow, 6 countries.
Yeah, it was a huge collaboration. In fact, a major funder, Wellcome, set up the Wellcome Sanger Institute in '92 specifically for this project. And the Sanger Institute actually ended up sequencing about a third of the entire human genome.
A third. Just one institute.
More than any other single contributor, yeah.
Yeah.
It really showed the power of that kind of focused, large-scale collaboration.
That level of international cooperation alone is incredibly impressive, especially for such a massive undertaking. It sounds like they had a pretty clear target from day one. But for something of this magnitude, what about the initial timeline and budget? Did they manage to stick to those ambitious projections?
Well, what's truly surprising, maybe, is that they didn't just stick to it. They actually exceeded expectations on both fronts.
Yeah.
Initially, it was planned for 15 years with a budget of about $3 billion, which was a huge amount back then.
Still is.
Right. But because of rapid advancements in the sequencing technology and really streamlined efforts across the labs, the project was declared essentially complete in April 2003.
So earlier than planned.
2 full years ahead of schedule. And the final cost was also less than expected, around $2.7 billion. Which, if you adjust for inflation, is maybe about $5 billion in 2021 dollars.
Still a massive investment, but under budget and ahead of schedule is quite something.
It really is. And what's particularly fascinating, I think, is the dynamic that was at play. You had this publicly funded project, the International Human Genome Sequencing Consortium, or IHGSC, running in parallel with a privately funded one, Celera Genomics, led by Craig Venter.
Ah, yes, the public versus private race. I remember hearing about that.
Exactly. This dual approach created this, well, synergy. There was collaboration, but also a healthy dose of competition, driving things forward incredibly fast.
That certainly added some drama to the science. But, okay, let's back up. To even think about sequencing 3 billion base pairs back then, it seems like an insurmountable challenge without some truly major technological breakthroughs. What made this project even feasible?
You've hit the absolute core of it. It really was a technological revolution. You have to remember, until the early 1970s, DNA was notoriously difficult to analyze, just isolating it, copying it, figuring out the sequence.
Yeah.
It was slow, painstaking work. The HGP was only possible because of a series of monumental technical breakthroughs in sequencing methods and related technologies.
Yeah. Okay, so 3 billion base pairs, that number is just hard to grasp. Can you maybe put that into perspective for us? How much data are we really talking about?
Yeah, it's tough to visualize, isn't it? Let's try this. Imagine the human genome is a massive, incredibly intricate book, written in a kind of secret language where each letter is a base pair. Before the HGP, we maybe had tiny fragmented paragraphs,
Mm-hmm.
and sequencing them was like trying to transcribe a sentence by hand from a giant library using just a magnifying glass.
Okay.
So 3 billion base pairs. Think of it like this. Imagine 3 big shipping pallets.
Okay.
Each pallet has 40 boxes stacked on it,
Right.
and inside each of those boxes are 5,000 pages of text.
Wow.
And each page is crammed, absolutely packed with 5,000 of those base pair letters.
Okay, that's staggering. That's an unbelievable amount of information.
It is, and accuracy was absolutely critical. So each fragment of DNA, each little piece, had to be sequenced roughly 10 times over.
10 times. Why so many?
To reach an accuracy level of over 99%, it wasn't just about reading the book. It was about triple-checking every single letter to make sure it was absolutely correct.
That's an incredible commitment to getting it right. So with all these technological leaps, how much faster did the process actually become?
Yeah.
Compared to what was possible before?
The speed increase was just mind-boggling. It really was. A testament to incredibly rapid innovation. Put it this way. In 1981, sequencing just 12,000 base pairs took about a year. A whole year of dedicated effort.
A year for 12,000. Okay.
And to 2001, just 2 decades later, you could do that same amount of sequencing in less than 1 minute.
From a year to under a minute?
Yeah. And at the peak of the HGP, these specialized sequencing factories were churning out DNA sequence at a rate of 1,000 nucleotides per second.
1,000 per second. What were the key technologies that enabled that leap?
Several things came together. Crucially, automated DNA sequencing technology. That replaced the really laborious manual processes. Then there was the widespread use of PCR, polymerase chain reaction. Think of that as like a super fast genetic photocopier.
Right, for amplifying small amounts of DNA.
Exactly.
Yeah.
Making millions of copies from tiny samples. Then, better cloning vectors were developed, especially things called BACs, bacterial artificial chromosomes. They were essential for handling really large chunks of DNA, sort of organizing the genome's vastness.
Okay, BACs. Got it.
And, of course, you needed much, much better computational methods, powerful computers and sophisticated algorithms to actually assemble the genome from all those billions of pieces.
That assembly part sounds like maybe the hardest challenge. Putting together a billion-piece jigsaw puzzle, but you don't even have the picture on the box lid. How did they manage that?
That's a great analogy, actually. They primarily used a method called hierarchical shotgun sequencing.
Mm-hmm.
So the whole genome was first broken down into smaller, more manageable chunks, maybe around 150,000 base pairs each.
Still pretty big chunks.
Relatively speaking, yeah. These were then inserted into those BACs, the bacterial artificial chromosomes. The BACs were put into bacteria, and the bacteria basically copied these DNA fragments as they multiplied. So you got countless identical copies of each fragment.
Okay, so you amplify each piece first.
Right. Then each of these smaller copied pieces was sequenced separately, using what's called a shotgun approach, essentially sequencing random bits within each piece. And finally, this is where the computational power was gone. Crucial algorithms were used to find overlapping sequences between all these fragments, like finding overlapping words in different copies of a shredded book, to piece them back together in the correct order and reconstruct the full chromosomes.
So it's like rapidly photocopying and translating almost every word of that massive book, then using computers to painstakingly piece all the pages back together.
Exactly. Page by page, chapter by chapter, until the whole book was assembled.
That really brings the method to life. So after all this groundbreaking work, this technological revolution, what were some of the most surprising discoveries? What came out of that initial draft sequence back in 2001? What did we learn about ourselves?
Well, one of the biggest surprises, something that really shifted our understanding of human biology, was the actual number of human genes. Before the project, estimates were all over the place, some as high as 80,000, even 140,000 genes. But the draft sequence revealed a much, much lower number, approximately 22,300, maybe 30,000 protein coding genes.
Wait, only 30,000-ish? That seems low. How does that compare to other organisms?
Well, that's the surprising part. It's a range quite similar to other mammals, like mice, which have around 30,000. But quite strikingly, it's even less than a simple mustard weed, Arabidopsis thaliana, That little plant has about 25,000 genes.
Hold on. Less than mustard weed. That definitely feels like a shock to the system. Does that mean we're somehow less complex than a plant, or or does our complexity come from somewhere else entirely?
Not at all. And that's precisely the profound insight, isn't it? What it really highlighted is that complexity doesn't just come from the sheer number of genes. It comes from how those genes are used and regulated. See, one human gene can actually code for multiple different proteins, sometimes hundreds, even up to 1,000 variations.
How does that work?
Through clever mechanisms like alternative splicing, where the gene sequence is kind of cut and pasted in different ways before it becomes a protein. Also variations in gene regulation, when and where genes are turned on or off, and modifications after the protein is made. So imagine a single gene blueprint, but you can build many different types of machines from it. That's how a relatively small number of genes can create such incredible biological complexity.
Okay, so it's not the number of blueprints, but the versatility of each one. That makes sense. And another big revelation you mentioned was about what used to be called junk DNA. What did we learn there?
Yes, junk DNA. That's another powerful example of how the HGP just shattered preconceived notions. We discovered that less than 2% of the human genome actually encodes genes in the traditional sense, the parts that directly build proteins.
Only 2%? So what's the other 98% doing?
Well, that leaves these vast stretches of non-coding DNA between genes. For a long time, scientists thought it was just junk. Evolutionary leftovers, maybe filler. But the project revealed that repeat sequences, for example, make up over 50% of the entire genome. And we now understand this junk isn't junk at all. It's absolutely essential.
How so? What does it do?
It plays crucial roles in regulating gene expression. Essentially, it's like the control panel, dictating when and where genes are turned on or off. It has significantly reshaped our genome over evolutionary time, providing what scientists now call an enhanced functional repertoire.
Mm.
It's like discovering the blank spaces in a book aren't empty, but contain complex instructions for how the story unfolds. It's the operating system for our biology, in a way.
The operating system. I like that.
Right.
So the junk is definitely not junk. What else did we learn about what makes us unique? Or maybe how much we're all alike?
This is maybe one of the most profound insights for humanity, I think. We discovered that almost all, a striking 99.9% of the nucleotide bases are exactly the same in all people.
99.9% identical.
Exactly. That tiny remaining 0.1% difference, though, that translates to about 3 million variations between any two individuals.
3 million differences. That sounds like a lot again.
It is and it isn't. These differences are mostly single nucleotide polymorphisms, or SNPs, tiny changes in a single DNA letter. They occur roughly every 2,000 base pairs or so. And these subtle variations are incredibly powerful. They're the source of all our individual uniqueness. They can relate to disease susceptibility, how we respond to drugs, or just variations in appearance or behavior.
So 99.9% the same, but that 0.1% holds all our individuality. That's fascinating.
It is. And another interesting tidbit, the mutation rate seems to be about twice as high in male meiosis compared to female meiosis, meaning sperm is, statistically speaking, the primary source of new mutations passed down through generations.
Interesting. Okay, so those tiny differences make us unique. What about how genes are organized? Are they spread out evenly?
Not at all. The genome landscape is anything but uniform. Genes aren't evenly distributed. Instead, you have these gene-dense urban centers, you could call them. These areas are typically rich in G and C bases, guanine and cytosine.
Okay, the GC-rich regions.
Right, and these tend to be more active regions.
Yeah.
Then you have gene-poor deserts, which are rich in A and T bases, adenine and thymine.
Like different neighborhoods in the genome.
Exactly. And you can even see these as distinct light and dark bands when you stain chromosomes and look under a microscope. And just for a bit of genetic trivia, chromosome 1 has the most genes, almost 3,000. While the Y chromosome, the male sex chromosome, has the fewest, just over 200.
That's a great overview of the early findings. It paints this picture of a shared blueprint with subtle but powerful individual variations. Now let's pivot to the promise this unlocked, this deep knowledge. It's fundamentally transformed medicine, hasn't it? Ushering in this era of personalized healthcare. How has that played out?
It really has. The HGP didn't just give us a map. It gave us the understanding to actually navigate the complex terrain of human biology. And that laid the groundwork for, well, unprecedented innovation in health and medicine. Critically, it enabled the development of what we now call precision medicine.
Precision medicine, tailoring treatments to the individual.
Exactly. Tailoring treatments to an individual's unique genetic makeup. This has profoundly increased the effectiveness of therapies. It helps minimize the trial and error approach and significantly reduces adverse drug reactions. It makes medicine far more targeted, far more patient-centric.
Can you give us some concrete examples? How is this actually impacting patients right now in the clinic?
Sure. Oncology cancer treatment is a prime example. We can now identify specific genetic mutations linked to different forms of cancer. This lets oncologists move beyond broad-spectrum chemotherapy, which kind of hits everything.
Right, with all the side effects.
Exactly. Now they can design therapies that target those precise mutations driving the cancer. This often leads to dramatically better outcomes with fewer side effects. And beyond cancer, we're gaining much deeper understanding of complex conditions like cystic fibrosis, Alzheimer's, heart disease. We can use genetic information to predict, with much greater accuracy, how a patient might respond to a certain medication. We're moving away from that one-size-fits-all model.
I can only imagine what this means for patients, especially those with conditions that were previously mysteries or had very limited options. What about rare diseases? They're often incredibly hard to diagnose, let alone treat effectively.
This is perhaps one of the most profound areas where genomic medicine is making a difference. There are thousands of rare diseases, maybe 6,000 or so currently identified, and about 80% of them have a genetic origin.
80%.
Yeah. Cumulatively, they affect millions. In the UK alone, it's estimated at 3.5 million people. Genomic medicine is revolutionizing their healthcare. It offers prompt, accurate diagnosis, sometimes ending years, even decades, of searching for answers, what they call the diagnostic odyssey.
Right, ending that odyssey must be life-changing for families.
Absolutely. And it allows for better risk stratification and, critically, the development of personalized treatments where maybe none existed before.
So, what if?
Mhm.
What if we could actually eliminate those diagnostic odysseys for rare disease patients? That seems like a huge potential benefit.
Exactly. Imagine a child with a mysterious, life-threatening condition finally getting an accurate diagnosis through sequencing, and then getting a targeted treatment based on that genetic cause. There's a powerful example, actually. A 10-year-old girl in the UK who had these severe, life-threatening reactions to chickenpox. Doctors were baffled for years. No. Whole genome sequencing identified specific mutations in her CTPS1 gene. This explained her vulnerability. But more importantly, it directly led to her being considered for a bone marrow transplant as a potential cure. That kind of precise, life-saving intervention just wasn't possible before.
That's incredible. Moving from complete uncertainty to a potential cure. And how is this genomic insight impacting cancer management beyond just diagnosis and initial targeting?
Well, cancer fundamentally is a disease of the genome. It's caused by accumulated mutations. So sequencing DNA directly from a tumor reveals the specific mutational landscape, the drivers of that particular cancer. This directly guides therapy. For example, drugs like vemurafenib target a specific mutation, BRAF V600E, found in many melanomas. The results can be significantly better than standard chemo.
Targeting the root cause.
Precisely. And beyond initial treatment, there's the potential of detecting circulating tumor DNA, or ctDNA.
DNA shed by the tumor into the bloodstream.
Exactly. Detecting ATT-DNA could let us spot cancer occurrence much, much earlier than conventional scans. This opens up a crucial therapeutic window of opportunity to intervene before the cancer progresses too far. It could really improve prognosis.
So moving towards more dynamic, real-time cancer management, another area you hear a lot about is pharmacogenetics.
Right. Pharmacogenetics is all about tailoring drugs to an individual's genetic profile. Genetic variations can predict how someone will respond to a drug, or, crucially, if they're at high risk for severe side effects.
Like drug allergies.
Exactly. Or other adverse reactions. For instance, in HIV treatment, a specific gene variant predicts the risk of a severe reaction to the drug abacavir. Knowing this beforehand allows doctors to choose a different drug before treatment even starts. It prevents harm and ensures safe, effective therapy. It's personalized prescriptions, essentially.
This sounds like it could revolutionize preventative medicine too, not just treatment. Can we use genetics to prevent diseases before they even start?
Absolutely. That's a huge area of potential. Take a condition like familial hypercholesterolemia. It's a genetic condition causing very high cholesterol from a young age. About 1 in 500 people carry a mutation for it, putting them at high risk for early heart disease.
1 in 500 is quite common.
It is. Genetic testing can identify these individuals early, even in childhood. Once identified, they can get aggressive lipid-lowering therapy, lifestyle advice, its primary prevention of cardiovascular disease, potentially saving lives decades down the line.
Intervening before the disease even takes hold.
Exactly. And the understanding from the HGP is also driving the development of completely new therapies. For example, there are phase 3 clinical trials for a drug called BMN 111. It shows promise in improving bone growth for kids with achondroplasia, the most common form of dwarfism. That's a direct therapeutic intervention based on understanding the genetic pathway involved.
That's a truly inspiring glimpse into the medical breakthroughs. It offers so much hope. But the HGP's impact isn't just confined to the clinic, is it? How has this knowledge spread into other areas?
No, definitely not. The insights have cascaded far beyond healthcare, reshaping really diverse fields. One crucial area is DNA forensics. Having the complete human genome map provides an incredibly powerful tool for law enforcement.
Right, identifying suspects or victims from crime scenes?
Exactly, from even minute biological samples with unprecedented accuracy. And, significantly, it's used by the military, like the U.S. Defense Department, to identify the remains of soldiers killed in combat using their DNA tissue banks. That brings invaluable closure to families.
So it helps solve mysteries, both criminal and historical. What about other sectors like agriculture or even environmental science?
The insights are remarkably transferable. In agriculture and livestock breeding, understanding plant and animal genomes has been revolutionary. Scientists can identify genes for things like drought resistance, pest immunity, or better nutritional content in crops.
So faster and more precise breeding.
Much faster. Developing new varieties of staple crops like rice or corn much more quickly than traditional methods. This is crucial for food security in a changing climate. Similarly, in livestock, genomic selection helps breed animals with better disease resistance or higher yields.
And environmental applications.
Yeah, microbial genome research is yielding fascinating stuff. By sequencing the genomes of bacteria and other microbes, we're finding new metabolic pathways. These could be harnessed for new energy sources like biofuels, or even for environmental cleanup, designing microbes to break down pollutants, for instance.
Tackling big environmental challenges using tiny organisms' blueprints.
Exactly. It's helping us understand life at its most fundamental level to solve large-scale problems.
It sounds like it's also fundamentally changing our understanding of humanity's past, our evolution.
It absolutely is. The HGP provided the ultimate reference point for comparing genomes. By studying genetic variations in diverse human populations, researchers can trace human migration patterns across the globe with incredible detail.
Following the footsteps of our ancestors out of Africa.
Precisely. Revealing those epic journeys. It also helps us understand key evolutionary milestones in a way fossils alone can't. We can see the genetic footprints of adaptation, population bottlenecks, even interbreeding events with archaic humans like Neanderthals.
Wow. And looking forward.
Well, the ability to identify heritable mutations helps us understand disease risk and potentially reduce their likelihood in future generations through things like genetic counseling and reproductive technologies. It's a form of preventative action on a population level.
It's clear the benefits are immense, truly remarkable. However, with such profound knowledge comes immense responsibility, doesn't it? It's not just about what we can do, but what we should do, how we manage this new power. What were some of the key ethical crossroads that emerged from the HGP? How are we navigating those?
You've hit on a critical point, and it was recognized right from the start. The HGP actually had a dedicated program, Ethical, Legal, and Social Implications, or ELSI. It was unprecedented for a large science project to build that in from day one.
ELSI.
Okay. And a commonly expressed fear, a really deep-seated one, was that this very personal genetic information could be used against individuals, particularly by employers and insurance companies.
That fear seems quite rational, really.
It wasn't just hypothetical. There were strong historical precedents showing how such information could be misused.
Can you give an example? Something that illustrates the real-world impact, maybe even before widespread genomic sequencing.
Sure. Take Tourette syndrome. It's a condition where individuals have normal lifespans. For many, it's mild tics that disappear. For others, it might involve motor and vocal tics, but they can often be managed effectively with inexpensive medication.
Okay.
Yet there are documented cases of employed, healthy individuals in their mid-20s managing their symptoms fine, being denied health insurance. The simple explanation on the denial paperwork, Tourette syndrome.
Just like that. based on the diagnosis alone.
Just like that, and organizations like the Tourette Syndrome Association report getting scores of such reports regularly. Insurers, for their part, worry about something called adverse selection.
Adverse selection.
The idea that if applicants know more about their future health risks from genetic tests than the insurer does, those expecting high costs might buy more insurance, skewing the risk pool. It creates a tension.
I see. That's a stark example. What about employment discrimination, especially for people who carry a genetic risk but aren't actually sick?
Right. The Americans with Disabilities Act, the ADA from 1990, does offer some protection against genetic discrimination for disabilities. But its scope is limited. It might not fully cover carriers of recessive genetic disorders, like sickle cell trait or cystic fibrosis carriers. They aren't ill, but they could pass the gene on.
So employers might worry about future costs.
Potentially.
Mm.
An employer might see hiring a carrier as a risk for higher future medical benefit costs, for dependents, perhaps, even if the individual risk is statistically small. It puts people in a difficult position.
And we know that genetic information, especially when viewed through existing societal biases, can unfortunately amplify prejudice. Troy Duster from UC Berkeley highlighted this very powerfully, didn't he, using historical examples.
He did. Duster pointed out that genetic information doesn't exist in a vacuum. It can ricochet against existing social structures and prejudices, often in harmful ways. He contrasted the experience of screening for Tay-Sachs disease with that for sickle cell anemia.
How did they differ?
With Tay-Sachs, screening programs within Ashkenazic Jewish communities were largely controlled by the community. They embraced the information, used it for voluntary counseling and reproductive choices, and it was very effective in reducing the disease incidence. The community managed the information.
Okay, a positive example of community control?
But the experience with sickle cell anemia screening in America was tragically different. It became heavily politicized, unfortunately framed as a disease of blacks. Mandatory screening programs were often imposed from outside the African American community.
And the information was misused.
Yes. Test results were sometimes used as a basis for discriminatory employment policies, denying jobs to healthy carriers. It highlights that critical what-if question that still haunts us. What if genomic data interpreted through biased lenses creates new forms of discrimination, a kind of genetic underclass?
How do we prevent that? Ensure the promise isn't overshadowed by the specter of eugenics.
It's a huge challenge. As Nancy Wexler from the ELSI group argued, the answer isn't to slow down the science. It's to make our social systems more accommodating, more just, and more resilient to this new knowledge.
That brings us squarely to another major ethical concern: privacy and control over our own genetic information. Who gets to see this blueprint?
Right. Privacy here means an individual's control over how their genetic information is generated, stored, and shared. Most people feel, quite strongly I think, that their medical records, including genetic data, should be strictly confidential between them and their doctor.
Seems reasonable.
But the reality is more complex. Genetic data is often obtained by third parties, employers, insurers, health, life, disability, even entities like the Medical Information Bureau, which serves the entire insurance industry. It raises big questions about consent and data ownership.
And it gets even trickier, doesn't it? When information about my genes can be revealed by testing my relatives, sometimes without me even knowing or consenting.
That's a huge ethical challenge. Genetic information can be deduced from relatives. Take Huntington's disease again, a devastating late-onset condition. A young adult at risk might want predictive testing for themselves, maybe for family planning.
Okay.
But if their test is positive, it automatically reveals that their at-risk parent also carries the gene. Whether that parent wanted to know or not, the information ripples through the family.
Right. The right not to know is also important.
Absolutely. And many people prefer not to know, especially if tests aren't perfectly accurate or if there are no effective treatments available yet. Remember, when free predictive testing for Huntington's was first offered, only about 10% of those at risk actually took the test. That speaks volumes about personal autonomy over this kind of knowledge.
So personal choice, familial implications, it adds layers of complexity. And this data isn't just sitting in individual files, is it? It's often collected in large databanks.
Precisely. Genetic data is increasingly stored in large, centralized databases. Think state forensic databases with genetic fingerprints from convicted felons, or the Defense Department's tissue bank for identifying military personnel remains.
Which raises questions about security and access.
Right. Yes, exactly. Who has access? For how long? Under what rules? Currently, there aren't really universal standards for protecting information in all these different kinds of data banks.
That's a complex landscape. Now, high pay, the Health Insurance Portability and Accountability Act from 96, was meant to protect identifiable health info. But we've recently heard about some concerning ethical lapses related to the HGP itself, specifically about where the reference genome actually came from.
Yes, that's a significant point that highlights the ongoing challenges around consent and anonymity. A 2024 investigation by Undark Magazine and Stat News revealed something quite surprising. It found that about 75% of the initial reference human genome, so the sequence everything else is compared against, actually came from a single anonymous donor's DNA.
75% from one person. I thought it was meant to be a composite.
That was the understanding. The consent forms reportedly stated no more than 10% from any one donor would be used to ensure anonymity and representativeness. The investigation also found that one of the project's lead scientists contributed about 10% of his own DNA to the reference.
Wow. That definitely raises questions about informed consent, doesn't it? And the whole idea of an anonymous reference genome.
It absolutely does. It highlights the inherent tension between individual privacy rights and the goals of public health or scientific research. It echoes past debates, like those around AIDS and mandatory disclosure versus individual rights.
These ethical considerations really underscore the critical role of genetic counseling. With this flood of data and complex implications, how is that field adapting?
It's a profound challenge for genetic counselors. The sheer volume of information is overwhelming. The field, which used to deal with a smaller set of known genetic tests, now really needs new subspecialties. No single person can realistically master all this rapidly evolving data. There are urgent questions about training, curriculum. Are the current two-year master's programs enough to prepare counselors for this complex new world?
And traditionally, counselors aimed for a non-directive approach, right? Just presenting facts, not making recommendations. Is that still feasible or even responsible?
That Joe Fridayism, just the facts, ma'am, is increasingly seen as difficult, maybe even untenable. Think about it. It's like a doctor saying, your cholesterol is dangerously high, it could kill you. Now you go figure out what to do. That's not how medicine works now. Doctors give strong recommendations.
Right.
With genetic data being so complex, often probabilistic and emotionally charged, it's incredibly hard to be truly non-directive. How you frame the information inevitably carries weight. The challenge is empowering patient autonomy while still providing meaningful guidance.
Makes sense. And I imagine there have been cases where that non-directive approach perhaps led to problems due to misinterpretation or maybe incomplete information.
There certainly have been challenges. An early example was the XYY karyotype controversy. Back in the 60s, flawed studies of prisoners wrongly linked the XYY chromosome pattern to violent or criminal behavior.
Oh, I remember hearing about that, the super male idea.
Exactly.
Yeah.
Even though those studies were later discredited, some counselors, years later, still presented XYY as carrying a risk of sociopathic behavior or learning disabilities. This forced parents into making huge reproductive decisions based on potentially misleading or just plain wrong information.
That's awful.
It powerfully highlights the need for counselors to critically evaluate information and present only what is truly relevant and accurate, avoiding misinformation or social bias. It's a constant tightrope walk.
Okay, so beyond the medical and ethical experts, what about the general public? How well do people understand genetics? Is public knowledge keeping pace?
That's a crucial question for the responsible integration of all this. A 2013 study in Durham, North Carolina, gave some interesting, maybe concerning insights. It found participants actually had pretty high factual genetic knowledge scoring around 84% on average, especially on things like disease inheritance.
That sounds surprisingly good.
On the facts, yes. But their perceived knowledge of the social consequences of genetic testing was significantly lower. Only about 64% felt they had some knowledge there. Compare that to 79% feeling knowledgeable about the medical uses. There's a gap.
So people get the science, maybe, but are less clear on the real-world implications, particularly the potential downsides.
Exactly. And the study showed concrete anxieties. Over half worried about DNA testing affecting health insurance. About 16% worried about job chances. This suggests a persistent gap between understanding the benefits and grasping the potential social and personal risks. Plus, all the media coverage, while raising awareness, can sometimes create an illusion of knowing. People recognize terms but don't fully grasp the implications, which hinders truly informed decision-making.
And maybe the way it's reported plays a role, focusing more on the breakthroughs than the potential problems.
That's a strong possibility. The imbalance in knowledge might well reflect a bias in reporting, emphasizing benefits over harms. It makes public education and engagement really challenging, but absolutely vital. Nancy Wexler, again from the ELSI group, had that wonderful analogy about this.
The movie analogy, right. ELSI writes the script.
Exactly. But the public must act as the producers. They need to help raise the money, hire the cast, get the movie made, and ensure its message is understood. Without that active public partnership, she warned, the movie will never be made in a way that truly benefits everyone.
Which leads to that profound what if? What if public understanding doesn't keep pace?
Right. Could misinformation lead to a backlash against genuinely beneficial uses?
Hmm.
Or conversely, could a lack of understanding allow negative consequences, like discrimination or privacy breaches, to go unchecked? Informed public engagement is paramount.
This deep dive really highlights the monumental scope, the impact, the ethical challenges. But the journey didn't just stop in 2003 when the HGP was declared complete, did it? The story kept going.
No, absolutely not. That term complete back in 2003 was relative. The HGP had sequenced about 92% of the euchromatic genome, the gene-rich, relatively accessible regions. The remaining 8% was mostly heterochromatin, really dense, highly repetitive stuff like the telomeres at the ends of chromosomes and the centromeres in the middle.
Tricky bits to sequence.
Exactly. Highly repetitive sequences are notoriously difficult for sequencing technologies to assemble correctly. They were like the missing chapters or maybe the complex diagrams in our genetic book.
So the main story was there, but significant gaps remained. How long did it take to finally fill those in, to get a truly complete sequence?
It took nearly 2 more decades, a real testament to continued innovation and sheer persistence. But in April 2022, a group called the Telomere-to-Telomere, or T2T, Consortium, published a truly complete sequence of the non-Y chromosomes. They finally filled that difficult 8% gap.
Wow, 2 decades later.
And then just last year, in August 2023, the Y chromosome was fully sequenced too. So now we finally have the complete sequence of all 24 human chromosomes, telomere to telomere.
A truly complete blueprint at last. Has that already yielded new insights?
Oh yes, this new complete reference genome has already identified over 2 million additional genetic variants, mostly in those previously unmapped regions. It's deepening our understanding of human diversity and the role these regions play in health and disease.
That's incredible, but it's important to remember, right, that the original HGP sequence and even this new one is fundamentally a mosaic, a composite, not one person's genome.
Precisely. It's derived from multiple individuals. It serves as a crucial scaffold, a reference point. The fact that we share 99.9% of it makes this mosaic incredibly useful for identifying individual differences in mapping disease genes. However, this brings us to a really critical ongoing challenge, the genomics diversity gap.
The diversity gap, meaning not all human populations are equally represented in these genomic databases.
Unfortunately, that's exactly right, and it's a major issue for global health equity. Despite the international nature of the HGP and many projects since, the overwhelming majority of sequenced genomes publicly available are still from people of European descent.
Why is that such a problem? Why is broader diversity so critical?
Because it creates dangerous gaps in our understanding. Genetic variations, susceptibility to certain diseases, how people respond to drugs, these can differ significantly across populations. If our knowledge base is primarily built on one group, then the benefits of precision medicine therapies tailored to genetic makeup might not work as well or might even be harmful for people from other backgrounds.
So it could actually worsen health disparities.
It absolutely could. It risks making personalized medicine a tool that benefits some populations far more than others, exacerbating existing global health inequalities. This leads to another urgent what if. What if we fail to bridge this diversity gap? Could the promise of genomics become concentrated, leaving vast populations behind? It's a fundamental challenge for inclusive science.
A critical issue we have to address. Now, on a more practical note, one of the most stunning impacts of the HGP has been the dramatic, almost unbelievable drop in the cost of sequencing.
It truly is staggering. It's perhaps the most tangible legacy. The cost reduction has outpaced even Moore's law for computing power. Think about this. Sequencing a full human genome cost around $95 million USD back in 2001.
95 million.
By 2008, it was down to about $342,500. Still expensive, but a huge drop. And by 2022, it had plummeted to an astonishing $525.
$525 for a whole genome. That's almost unbelievable. It's practically a routine lab test cost.
It's getting there. This exponential decrease is fundamentally democratizing access to genomic information. It's moving from an elite research tool towards something potentially accessible for routine diagnostics.
So what if? What if that cost continues to fall? What if personal genome sequencing becomes as common, as accessible as getting a blood test? How might that transform everyday healthcare?
That would be a paradigm shift.
Yep.
It would fundamentally transform healthcare towards being truly proactive and predictive.
Mm.
We could move from just reacting to illness toward highly individualized predictive diagnostics, tailored preventive strategies, all on an unprecedented scale.
Like having your genetic blueprint guide health decisions throughout life.
Potentially, yes. From personalized nutrition and exercise plans, to identifying risks early and mitigating them, to ensuring medications are perfectly tailored to you from the start. And this whole field, sparked by the HGP, has already been a massive engine for economic growth. It's
Yeah.
created new biotech industries, fueled drug discovery, generated countless jobs in R&D and clinical application. It keeps expanding.
That vision of a future shaped by genomics brings us back to Nancy Wexler's call for public partnership. ELSI writes the script, but the public produces the movie.
Exactly. We need an informed, engaged public as partners every step of the way to ensure that this incredible scientific progress translates into equitable societal benefit, not new forms of inequality or discrimination, issues around fairness, privacy, access, how the information is used. They're still very much alive and evolving as the science races ahead. Laws like GILINA, the Genetic Information Non-Discrimination Act in the U.S., try to address some concerns, but the legal framework often lags behind the technology. So continuous public awareness, education, and thoughtful societal dialogue are absolutely vital to navigate these complexities responsibly. We need to collectively shape this future.
We've certainly navigated a complex landscape today, from the HGP's ambitious beginnings, those incredible technological leaps, to its profound impact on medicine and these deep ethical dilemmas it unveiled. It's a story of, well, unparalleled scientific triumph. It offers unprecedented insights into what makes us, us. The HGP didn't just map our genes, it really mapped a new frontier for humanity. We now have this profound understanding of our shared genetic heritage and also the subtle variations that make each of us unique. But as we've discussed, this knowledge comes with immense power and immense responsibility. So as you reflect on this journey through our genetic code, here's something to consider. What if, armed with the complete blueprint of human life, we could not only cure disease, but potentially enhance human capabilities, design future generations even? Where do we draw the line between healing and enhancing? And maybe more importantly, who gets to decide? The deep dive into our genetic future, it has only just begun.